Welcome to MycoTalks, which is hosted by the MRC Center for Medical Mycology at the University of Exeter. My name is Judith Berman, and I'm from Tel Aviv University in Israel. The co-chair today is Christina Cuomo from the Broad Institute in the United States. Today, our speakers are Alessia Buscaino from the University of Kent in the UK and Sing Wei from Cincinnati Children's Hospital Medical Center in the United States. MycoTalks are organized by the committee shown at the bottom of this slide with valuable assistance in running the event from MRC Center staff. MycoTalks are held at 1600 UK time on the last Thursday of every month. They were started by the MRC back in July 2020 to bring together the medical mycology community during the COVID-19 pandemic to hear about current science advances when we could not attend face-to-face conferences. Even though the pandemic is less of a concern, MycoTalks attracts a global audience of around 300 scientists, with many more who view the recorded talks later on YouTube. Apart from great science, the philosophy of MycoTalks is to promote diversity in our field in terms of career stage, gender, geographical location, and research field. And the committee is always happy to receive speaker suggestions for our future program. The format is two 30-minute talks on different aspects of medical mycology, followed by a 30-minute question and answer session at the end. To ask a question, please post it in the Q&A tab, which will be seen only by the moderators. It is important to state the name of the speaker you wish to address. The chairs will alternate the questions between the speakers and may choose to amalgamate questions that cover a similar topic. However, even if we run out of time, all questions posted to the speakers on the Q&A will be sent to them after the event. The upcoming MycoTalks are listed on our website, and you must register for each session. Please note that the time of the talks is 1600 UK time, so in the spring and autumn, when daylight savings time status shifts, please check how the timing might affect your time zone. We would like to draw your attention to the Medical Mycology Trainee Seminar Series, which also is held monthly and provides a great opportunity for early career scientists to give a talk. More recently, we also launched the MycoClinics series of online talks that have a specific clinical focus. Again, you can find the schedule for these on the MRC Center website. So now I will introduce Professor Alessia Buscaino, a group leader at the Kent Fungal Group, University of Kent in the UK. Alessia grew up in sunny Sicily, where she graduated in molecular biology at the University of Palermo. She did her PhD with Dr. Asifa Akhtar at the Embol Labs in Germany, where she developed her interests in non-coding RNAs, chromatin modifications, and epigenetics while investigating mechanisms of dosage compensation in fruit flies. For her postdoctoral studies, Alessia moved further north to the laboratory of Professor Robin Alshire in Edinburgh. Thanks to an EMBO long-term postdoctoral fellowship there, she investigated the interplay between RNAi and heterochromatin in the fission yeast Schizosaccharomyces pombe. Keen to apply her expertise in model systems to clinically relevant pathogens, Alessia came to my lab in Tel Aviv, Israel for a short-term EMBO fellowship to start exploring how non-coding RNAs and chromatin regulate the biology of the major fungal pathogen Candida albicans. Excuse me. Alessia joined the University of Kent in 2013. Her group is dedicated to understanding how chromatin and non-coding RNA affects fungal pathogen adaptation to different host niche environments. In addition to her scientific discovery, Alessia is passionate about diversity and inclusivity in science and higher education. The title of her talk today is Discovery of an Active RNAi Pathway in Candida albicans. And Alessia, please turn on and share your screen.
Yeah,
and I'll remind you, I can hear you. And We can see your screen. And I want to just remind the audience to post questions in the Q&A with the name of the speaker that they are intended for. Thank you very much.
Thank you, Judy. I hope you can all hear me well. It's a pleasure to be here, and thank you, Judy, for the introduction. And yeah, today I will discuss with you our latest finding on RNAi in C. albicans. So, fungal organisms, I think we all have our reason why we love fungal organisms. And personally, one of the things that I found the most astonishing about fungal organisms is that they can adapt very rapidly to very different environments. And this allows them to thrive in so many different microenvironments, such as, for example, the microenvironment they are found in the host, but also the different environment found in the wild. And the fungal microorganism can also interact with other macros. C. albicans, for example, is a commensal organism that colonizes the GI tract of the majority of us without creating any problem. However, C. albicans can also become a dangerous pathogen, and when it does so, it migrates from the GI tract and colonizes vital organs, causing diseases that are associated with a large number of deaths. C. albicans can also withstand antifungal drug treatment. And C. albicans adopt a battery of virulence factors and fitness attributes that drive this adaptation. And because of their importance, it's not surprisingly that those virulence factor and fitness attributes are very well studied. For example, morphological plasticity. C. albicans is able to grow in different morphological forms, and this switch is important for virulence, but also genome plasticity. C. albicans is able to shuffle its genome, and this is allow for selection of genotypes that are better fit to grow in a new environment. RNAi interference is actually a very ancient adaptive mechanism. And in the next couple of slides, I will explain to you, first of all, what RNAi is and why I think RNAi is a great mechanism for driving adaptation. So, what is RNAi? RNAi is triggered by double-strand RNA molecules, and those that can be made from outside the cells here, or they can be made in the cells. And they can be made in the cells thanks to transcription. Indeed, the transcription of a genomic region can lead to an RNA that can fold itself, creating a double-strand RNA, or convergent transcription can lead to double-strand RNA. Those double-strand RNA are recognized by the RNAi machinery. And this is at its simplest form, is formed by two proteins, dicer and argonaute. Dicer recognized the double-strand RNA and chopped it into small interfering RNA of 21 to 23 nucleotides. And one of these RNA strands is bound by argonaute, and argonaute can then target complementary nucleic acids to bring about three main functions, really. Gene silencing, RNAi can promote genome stability, and RNAi can also modulate interaction with other organisms. And I just wanted to share to you why I think RNAi is actually an ideal mechanism for adaptation. And this is really because its trigger is a double-strand RNA molecule. And because of this, RNAi, by definition, is a plastic mechanism because it can be modulated by transcriptional changes. So transcriptional changes can change the amount and the nature of this double-strand RNA, and therefore change the RNAi pathway. RNAi is also very rapid. It is estimated that RNA polymerase II can transcribe between 1 to 3 kb every minute. And RNAi is also reversible because it depends on transcription. It's possible to switch RNAi on and off by switching transcription on and off. So I think if we consider these three main features of RNAi, it's probably not surprising that many organisms, including fungi, have used RNAi for adaptation. And I'll just give you a few examples here. For example, in mucor, RNAi-directed gene silencing is important for development of antifungal drug resistance. Recently, it has been shown that in Cryptococcus, RNAi regulated transposon activity, and this again is important for development of antifungal drug resistance. And pioneer work with plant fungal pathogens have shown that the small interference RNAi are actually mobile, and they can be transferred between organisms, and this trans-kingdom RNAi can regulate pathogenicity. So, considering the importance of RNAi across the fungal kingdom, the question really is, why has RNAi been overlooked in C. albicans? And the first question I wanted to really address is, can Candida albicans have an active RNAi pathway? And this question is not really trivial, because actually it is known that RNAi has been lost several times during evolution. For example, Cerevisia does not contain an AGO gene or a dicer gene, and therefore does not have an active RNAi pathway. While Castelli, a close relative of Cerevisia, has an AGO gene, has a dicer gene, and therefore has an active RNAi pathway. So what about Candida? So if you look at the Candida clade, some member of the Candida clade, but not all of them, have a gene encoding for Argonaute, and this is included Candida albicans. So Argonaute in C. albicans encode for a canonical Argonaute protein. C. albicans also encode a gene for a non-canonical dicer protein. But when we started this project, actually, it was really not really clear whether C. albicans had an active RNAi pathway or it didn't. And in the next couple of slides, I will explain to you why this was not clear. So first of all, the Bartel and Fink lab, almost 15 years ago now, they used a wild-type strain that was coming from the reference strain for the C. albicans community, S4E5. And they used this strain to sequence small interference RNA in C. albicans. And by doing so, they could identify sRNA hotspot in C. albicans. However, they did not go further to assess whether those sRNA are functional or not functional. Following up on this paper, the White lab decided to use again the same strain to develop a synthetic RNAi system where they construct a gene construct that could transcribe an RNA molecule that was folding as an hairpin. And this RNA molecule was complementary to another tumor gene. And the idea was relatively straightforward. This RNA molecule should trigger an RNA response and therefore silence the other tumor gene. However, when they done these experiments, they could see no silencing. And therefore, the question really at that point was, is RNAi functional? And this is really the question that we try to address in this project. We are actually interested in understanding the role of RNAi in the whole Candida clade, but we decided to start on C. albicans simply because there was some previous work done. So how do we go about to address this in the most comprehensive way as possible? So our assumption was that S4E5, the wild-type reference strain, had an active RNAi pathway. And if this was the case, it should be possible to identify the region of the genome that contain sRNAs. And we reasoned that if we delete Argonaute, we should kill RNAi activity, and this should lead to upregulation of RNAi target gene. So we decided to do this simple experiment. We create an AGO1 deletion strain by CRISPR-Cas9 in the C534 background and sequence long and short RNA by sequencing. However, when we did this experiment, the results really demonstrate that the C534 and therefore C. albicans RNAi was not active. And this is because although we find sRNAs in wild-type cells, those sRNAs, the genes that contain these sRNAs are not upregulated in AGO deletion cells. In other words, there is not an overlap between SRNA hotspot and a hyporegulation of AGO deletion in AGO deletion strain. So we were a bit disappointed, but we revised our hypothesis, and now our new hypothesis was that RNAi actually is inactive in C. albicans. However, we decided to try to understand why this was the case. So we go back again to the core RNA machinery. So as I was saying, there are two key proteins for RNAi. One is dicer and one is argonaute. And then we excluded straight away that dicer could be inactive in C. albicans because, again, the Bartel and Fink lab have shown that expressing C. albicans dicer in Castelli could restore RNAi activity. And therefore, we knew that C. albicans dicer was an active protein that is able to do RNAi. And so we focused our attention on argonaute. And the Candida genome contains one AGO gene, AGO1, that encodes for a protein that is a canonical argonaute protein. It contains an N domain, a PAS, a MIDI, and a PIWI domain. And when we look at key amino acid residues in this domain, we noticed that C. albicans argonaute contain a change in a key residues. E361 in C. albicans is a lysine, is a K. And we strongly suspect that this was the reason of RNAi inactivity, because the corresponding mutation in worms or in flies completely kills RNAi activity. And actually, by serendipity, this mutation, E2K, was the mutation that led to the discovery of argonaute in the original RNAi screening in worms. So we strongly suspect that this was the reason why C. albicans RNAi was inactive. Now, all these experiments were done with a C531.4 strain that was in our lab. So it was a possibility that actually was our strain that was mutated and not the C531.4 across the world. So to address this question, we teamed up with Juliana and Christophe at the Pasteur Institute, and we decided to look at the sequence of argonaute in a panel of 25 strains that were coming from different labs across the world. And we also look at the sequence of argonaute in vivo P17 and SN150, two strains that come from C531.4. And as you can see here from this sequence alignment, all the strains that we looked at, they should be RNAi inactive because they have the inactive K residues in a position 361. As a reference here, you see how Drosophila argonaute 2 has an E at this position. Therefore, going back to the reason why there were these apparent conflicting data in the literature when we start the project, we think this is simply because although dicer is active and can create small RNA, this small RNA cannot silence marker gene because of a mutation in argonaute. The next question we really want to ask whether the reference strain in C. albicans is representative of the species. In other words, we wanted to know whether AGO was mutated also in other clinical isolates. So first of all, what we've done, we look at argonaute sequence in W1, another C. albicans clinical isolate. And as you can see here by the Sanger sequencing, W1 should have an active RNAi machinery because of the active E residues. So the question really at this point was which one is the odd one out, W1 or C531.4? And to answer this question, again, in collaboration with Christophe and Juliana de Pasteur, we look at a panel of 295 clinical isolates. So what I'm going to show you here in a second is a heat map where we are focusing on the PAS domain, and I'm highlighting here all the residues of the PAS domain that are conserved between C. albicans and other organisms. And here with the arrow, I am highlighting the residues K361. So what are the results of this in silico analysis? So first of all, we discovered another isolate that, similar to a C531.4, is supposed to be inactive because has a KK homozygous mutation. We find also 8 other isolates that they are heterozygous for this mutation. They are EK. While, and most importantly, the vast majority of the other clinical isolates that we looked at, they have an active EE residues, and therefore should have an active RNAi machinery. We perform a phylogenetic analysis to see whether the strain with the mutation are related, and the answer is most probably yes. The majority of them, but not all of them, they actually cluster in the same part of the phylogenetic tree. And therefore, we think that this mutation was selected somewhere here in the ancestor of these strains. Okay, so what do we know so far? We know so far that the reference strain C531.4 is not representative of the species for RNAi, because the HAGO E61K variant, so the inactivating variant, was not selected in a lab. It must have been selected in the wild. And of course, the question is there in selective pressure that gives an advantage in selected for this mutation? And we don't know the answer to this question yet. And we also have shown that the vast majority of C. albicans isolate are predicted to be RNAi active. Of course, at this stage, we did not really show that RNAi was active in the other C. albicans isolates, and this is what we wanted to do. So what we done? We took an approach very similar to the one we've done before. We focus on a panel of isolates that they are rated to be active. And our hypothesis was that, again, if RNAi was active, we should be able to identify the coding and non-coding region of the genome that have sRNAs. And we decided in this case to create an RNA-specific dicer mutant, and this should kill RNAi activity and therefore lead to upregulation of those RNAi target genes and ablation of small RNA. We also generate an Argonaute mutant to kill again RNAi activity, and this should lead to upregulation of RNAi target gene. And to address this question, we perform again a long and short RNA sequence and look for coding and non-coding genes that had sRNA in wild type, and those sRNA should have been bound in dicer, and the long RNA corresponding to this sRNA loci should be upregulated in both dicer and Argonaute. And to cut a long story short, we find actually just one set of genes that meet this criteria, and these are the TLO genes. So what are the TLO genes? This is a family of C. albicans genes. The majority of them are found as subtelomeric region, with the exception of one that is found in the arm of chromosome 1. And TLO genes encode for a component of the mediator complex. And several studies from the Anderson, Berman, Moran, and Sullivan lab have shown that TLO genes are important for virulence and for adaptation. So in the next couple of slides, I will show you the data demonstrating that TLO genes are RNAi targets. So first of all, we find sRNA against TLO genes here in wild type, and those sRNA are more or less of the right size to be dicer products. And more importantly, these sRNA are gone in a dicer mutant. Secondly, as a bona fide RNAi target, a TLO long transcript are upregulated in dicer RNAi strain and Argonaute deletion strain compared to wild type. And complementing the HAGO mutant with a wild type version of Argonaute rescue this upregulation. So therefore we can conclude now that C. albicans RNAi is active and TLO genes are a target of RNAi. However, at this point, we have not shown whether the E361K is responsible for the inactivity of RNAi in the reference strain. And we addressed this question a couple of different ways, but here I'm going to show you one approach that we took. And the approach was the following. We decided to cure RNAi in S4E5 by reintroducing the active E residues at position 361. And we perform long and short RNA sequence of this new strain and compare to the wild type one. And as you can see, once we do that, the results were really clear. We now see down-regulation of TLO transcript when we cure RNAi, demonstrating two things. Again, confirming that TLO genes are RNAi target, and secondly, that it's possible to cure, to reverse RNAi inactivity in S4E5. So going back to the original question, why has RNAi been overlooked in C. albicans? Well, it has been overlooked because the reference strain S4E5 is an RNAi mutant. And in contrast to S4E5, RNAi is active in other C. albicans clinical isolates, and RNAi silence subtelomeric TLO genes. The people that done the work, everybody in the lab, with special thanks to Elise, that is a postdoc funded by the Leverhulme Trust, that done the majority of the wet lab work for this project. And Christina is a postdoc funded by the BBSRC that done the bioinformatic analysis. As I was saying, for this project, we are collaborating with Rebecca, Christophe, with Juliana, and Corinne and Christophe's lab. I need to thank everybody else in the lab, all collaborator mentor across that always helped us during the year, and everybody in the Kent fungal group. And thank you all for listening.
Thank you, Alessia. Please post your questions for Alessia in the Q&A, and we will come back to them at the end. Our second speaker today is Dr. Sing Wei. Dr. Wei has made important contributions to reproductive and developmental immunology with direct relevance to improving pregnancy outcomes and the health of infants. His lab has pioneered tools for tracking immune components with fetal specificity and utilized these tools to reveal fundamental insights on how pregnancy immunology unfolds. This includes distinctions between immune tolerance versus rejection, driving the pathogenesis of pregnancy complications, and how mothers immunologically remember their babies as a basis for how pregnancy impacts the susceptibility of women to complications in future pregnancies. His group showed also hyporesponsiveness for neonatal immune cells reflects of active suppression as opposed to immaturity, and the importance of this suppression allowing babies to tolerate their first commensals. Dr. Wei is the founding director for the Center for Inflammation and Tolerance at Cincinnati Children's Hospital and the principal investigator for the March of Dimes, Ohio Collaborative on Prematurity Research. And his leadership in these areas has been recognized by the HHMI and the NIH Director's Pioneer Award, among other funding. Welcome to MycoTalks. Dr. Wei. Sing, can you please share your slides?
Thank you so much, Christina. Do the slides look okay?
Yes. Looks
okay.
Good.
So, as Christina said, I don't consider myself a fungal expert, and so I'm super excited to talk with you guys today about some of the work that we're trying to do by studying fungi and to teach us maybe some fundamental aspects about how our bodies work, and particularly how our immune systems work. The way that Christina introduced me, I think you would probably be wondering, what is a reproductive developmental pediatrician? Why would he be studying fungi? And I just want to walk through some of this reasoning for how we became interested in this topic. First is from the perspective of commensalism that Alyssa already outlined. Our bodies are filled with trillions of microbes that colonize our mucosal barrier tissues like the intestine. And how we tolerate these microbes, is really unknown, in that why our bodies don't mount a inflammatory response to this huge repertoire of extended self-antigens expressed by the commensal microbes. And in many ways, that's the fundamental question in reproductive biology, why the mother doesn't mount a inflammatory response to the developing fetus that also contains a huge repertoire of paternally expressed, paternally encoded extended self-antigens. And so that's one touch point for why a reproductive biologist would want to study fungi through the lens of commensalism. And I would say another touch point is the idea that if we wanted to study commensalism, the majority of what's known about how our bodies tolerate commensal microbes is really through the lens of 16S ribosomal sequences. And so microbes like viruses or fungi that don't express 16S are essentially invisible to that platform of analysis. And so we thought that if we studied how commensalism to fungi works, and if we could find that process is similar or identical to bacterial commensals, that's one thing. But even more excitingly, if we can find differences in how our bodies tolerate non-bacterial or eukaryotic commensals like fungi, then that would be even more exciting. And we started on this type of reasoning about 10 years ago now. And in addition to this type of reasoning, one of the most important aspects of it is we had to convince somebody to do it. And this is Tony Zhang, who at that time was starting the combined MD PhD program at the University of Cincinnati. And he decided to join my lab that was a reproductive and developmental immunology lab, but instead of studying pregnancy and newborn babies, he was going to study commensalism through the lens of fungi. And one of the first things that Tony discovered was that people had already known that it's important for us to have commensal microbes because if you looked at mice that were raised in notobiotic germ-free facilities that were lacking commensal microbes, their immune cells were very, very different and very, very skewed. And they had a lot of immunological defects, including being super susceptible to aberrant intestinal inflammation in the form of colitis, or inability to mount normally protective immune responses that made them susceptible to all sorts of different types of infection. And around that same time, Ken Caldwell's group had shown that it wasn't just the commensal bacteria that were important, but that commensal viruses could totally replace commensal bacteria, because he monocolonized germ-free mice with viruses and showed that many of those immunological perturbations that were found in those germ-free animals were overturned upon virus colonization. And so we basically asked a simple question is that, well, if commensal microbiota in the form of bacteria are important, and if viruses can replace bacteria, can fungi also replace bacteria? And that's what one of the most important things that Tony showed is that fungi can absolutely do it, because if we establish monocolonization of mice with Candida albicans, then many of those detrimental phenotypes that people had reported in germ-free mice were overturned. So those mice became less susceptible to intestinal inflammation in the form of colitis, and then their immune cells were tonically stimulated to allow them to very efficiently protect against infection. And so, at least at a first pass, what this meant to us was that in many ways, there's a lot of functional redundancy between bacterial commensals and non-bacterial commensals, especially viruses and more importantly, fungi in the form of Candida albicans. Next, we pivoted to ask, well, what happens when fungi and bacteria co-colonize together? In the prior ones, it's really monocolonization between each of these types of commensals, but in a more realistic setting, these things coexist together in our mucosal tissues. Then this was carried on by another graduate student, Jenny Xiao, who showed that when they co-colonize together, the Candida or fungi drive a very selective immunological response that I call Th17 inflammation. And the way that Jenny was able to show this, is that she took mice and established long-term colonization with recombinant Candida albicans that was engineered by Judy Berman to express model antigens, including a model antigen peptide called 2W1S. And we use tools generated by other people to track these CD4 cells with this specificity with really high precision. And we found that in mice that are colonized with these recombinant Candida, there was a pretty massive expansion of peripheral CD4 cells that recognize the colonization. And that a lot of these cells then differentiated into what we call Th17 effectors because they express the transcription factor RR gamma T. And the presence of these Th17 effectors in mice that are colonized then protects the mice against invasive fungal infection. Because if we challenged or intravenously infected these mice that were colonized, then they were highly resistant to Candida infection. What was really interesting to us is that tonic stimulation by fungi with driving Th17 immunity not only protected the mice against Candida infection, but they protected mice against other infection by other extracellular pathogens that Th17 immunity was important for protection. Here's an example of Staph aureus susceptibility in mice that are colonized with Candida and red. After infection with Staph aureus, none of the mice die, whereas control mice without Candida colonization, the majority of them succumb shortly after infection. And when they succumb, they're chock full of Staph aureus bacterial burden in their target tissues, like the kidneys, whereas colonized mice have sharply reduced bacterial burdens. I think another point that was really interesting with this data is that we could see if tonic stimulation by the fungal colonization was essential. And this was shown by then treating the mice with fluconazole to then eradicate the fungal colonization. And what Jenny showed is that tonic stimulation is absolutely essential, because if you eradicate fungal colonization, even though mice were previously resistant to infection, then they susceptibility recurs. That's associated with a loss of Th17 immunity. And the presence of Th17 immunity not only protects against infection by Candida and other extracellular pathogens, but also has a negative consequence in that these animals are also much more susceptible to aberrant airway inflammation, and especially triggered by Th17 or neutrophilic triggers. And so a lot of that is summarized in this graphical abstract where we showed co-colonization with fungi and bacteria drive expansion of fungal specific cells that produce IL-17 that then in turn activate circulating neutrophils that protects not only against Candida infection, but also against Staph aureus infection with a negative inflammatory consequences. Okay. Well, the next question for us is then, well, how does fungi do this? As the first speaker already also described, Candida can exist in different morphological states. And one of the key transcriptional regulators for controlling morphological changes is something called UME6. And so we wondered which morphology during colonization was the most important for driving this expansion of fungal specific Th17 cells. And what was really interesting to us is that when we locked the Candida into a UME6 null state, or if we force constitutive expression of UME6, in neither situation, we found that systemic priming of Th17 immunity. But it was only when we forced Candida to oscillate between high and low UME6 expressing states that we found this priming of Th17 immunity, which suggests that when fungi are colonizing the intestinal tract, they're not locked into a fixed transcriptional state, but instead it's dynamically controlling morphological changes. And it's that dynamic changes that's driving immunogenicity that drives this systemic immunological response. Okay. Well, then this led us to ask, well, how is the presence of this commensal fungi tolerated in the host intestine? Because if they're driving a big Th17 pro-inflammatory response, then why is it that our intestines or other mucosal surfaces that are colonized with fungi don't have constant inflammation? And again, we're tracking the systemic accumulation of CD4 cells with this surrogate fungal specificity, because we're colonizing with recombinant Candida that expresses this one model antigen 2W1S. And when we started tracking the cells, I've already told you that a lot of them differentiate into this Th17 program, and less into other defined CD4 differentiation programs. But what was also really interesting for us is that the majority of these cells that are primed by commensal colonization actually didn't belong to any known CD4 differentiation lineage. And so we were trying to figure out then what they did differentiate into. So we sort purified these cells and subjected it to single cell RNA sequencing. And somewhat interestingly, we found that the majority of these cells actually express a transcriptional regulator called Kreppel-like factor 2 or KLF2. And turns out this KLF2 was really important for maintaining allowing our bodies to maintain tolerance to commensal fungi, because if we took mice that KLF2 was conditionally deleted in the CD4 cells, then a majority of these mice spontaneously developed rectal prolapse in our animal facility. And we knew that this was driven by commensal microbiota, because if we treated the mice with antimicrobials to eliminate the commensal microbiota, then they didn't develop rectal prolapse anymore. And when these mice got sick, with intestinal inflammation and rectal prolapse. Their mesenteric lymph node that drains the intestines are chock full of these ROR gamma positive or Th17 cells that are outlined in this red. So these KLF2 cells that are being primed actively suppress inflammation caused by these otherwise pathological Th17 cells. And we think this may be really important to understand not just how commensal tolerance is regulated, but human disorders that are associated with aberrant tolerance to commensal microbiota, like inflammatory bowel disease. Here's our analysis of publicly available single-cell sequencing data from intestinal biopsy specimens from patients with Crohn's disease compared with healthy controls. And what you can see is that intestinal biopsy specimens from Crohn's disease patients selectively have reduced KLF2 cells. And this reduction is really prominent mostly in the non-defined lineages of cells, so non-Tregs, non-follicular helper cells, whereas there's very little KLF2 expression in traditional regulatory T cells. Okay. And this is in sharp contrast to another known immune suppressive cell type called regulatory T cells, because in these samples of patients with Crohn's disease, there's no defect in accumulation of traditional Tregs identified by FOXP3 expression. Whether you look at all CD4 cells or a more selective gate excluding T follicular helper cells and Tregs, or focus just on the regulatory T cells. And so because of that, a few months ago, we published this paper that suggests these regulatory T cells that people have largely implicated in commensal tolerance may have a really important role in self-tolerance, and the intestinal inflammation that people report may still be defects in self-tolerance in mucosal tissues, but highlighting what we think is a really important for another lineage of CD4 cells identified by this transcription factor, Kruppel-like factor 2, that may be important for sustaining commensal tolerance. Okay. And then last minute, I'll just say that we're trying to bring this back now to take this information that we're learning for how fungi do it, then to apply it to reproductive tolerance by asking if those KLF2 CD4s that are really important for maintaining fungal tolerance, how would they impact fetal tolerance during pregnancies, and how does baby's first colonization differ from colonization that's acquired in later postnatal life, again using a lot of these similar microbe and immunological tools. Okay, and lastly, I just want to highlight the people that have worked with me on this project. Tony, like I said, was the first MD PhD student who set up this platform in my lab. And then a lot of the subsequent work was carried out by Jenny Shao. We've had outstanding internal collaborators in Teresa, Hitesh, and Artem. And then coming into the fungal immunology field, I found that this has been the most welcoming field in terms of people who have shared their expertise so generously, and especially Richard Bennett at Brown, who has taught me all things fungi, and Judy Berman, who made the original recombinant Candida albicans that we've used for our studies. So thank you so much.
Thank you very much, Sing Sing. And please remember to get your questions into the question and answer tab. We'll be starting our question and answer session now. And make sure again to put down the name of the speaker whose question your address, whose talk you're addressing with your question. So I'll start with with a question from Priyesh Singh, who asks, why would inactive AGO1 have been selected in Candida albicans SC5314? And that question is for Alessia.
Yeah, thanks. So the short answer is I don't know, but there are a few things I think would be good to highlight here. It's been selected for sure somewhere in the host, not in the lab. So, yes, and we just found a second one, clinical isolate, that is related to SC5314. So it could be that there is not a selective advantage. However, I don't know if you have noticed, we actually seen an heterozygous mutation that was a bit far away from the phylogenetic trees, and this suggests that perhaps there is a selective pressure. We do really just need many more inactive RNAi mutants to really be able to address this question properly. At the moment, we just have 2 out of 300. It's very difficult to really have a cause-effect relationship. But yes, definitely we would like to know.
Okay. Christina, would you like to address a question to Sing Sing?
Sure. We have a question, Sing Sing, from Gordon Brown, who comments, very nice talk indeed. Do you have an idea of how the KLF T cells are suppressing the responses?
Yes, thank you so much, Christine and Gordon. Yes, so I didn't get a chance to describe that, but turns out the KLF2 cells are really proficient at making immune suppressive cytokine called IL-10, and IL-10 production by them is essential for averting colitis. Because if we take mice that don't have KLF2 cells and transfer in wild type KLF2 cells, we can ameliorate colitis. But if we transferred in IL-2 deficient, IL-10 deficient KLF2 cells, then we don't get that reversal of colitis. And I would just say that IL-10 production by FOXP3 negative cells is highly controversial. People have labeled them sort of globally as these TR1 cells or T regulatory type 1 cells. Even though KLF2 cells produce a lot of IL-10, they're by no means the only cells that produce IL-10 that are FOXP3 negative. And when we look at that, it's probably the minority, maybe a quarter or a third of the IL-10 is produced by the KLF2 cells. So there's still tons of other IL-10 producing cells out there, but I would say that our data would suggest IL-10 production by the KLF2 cells is probably the most important for averting colitis.
And then the second part of this question was you also showed an interaction or suppression of Th17. Is there a effect on other cells that you, I guess you're, yeah, can you just comment more on these types
Sure.
of, yeah.
Sure. So thanks for that question too. So when we pull out the KLF2 cells, so when we sort purify them and use them in classical suppression assays that people had used for other suppressive cell types like regulatory T cells, they're just as potent as classical FOXP3 cells in terms of suppression. And in those assays, they suppress proliferation of responder cells no matter how you stimulate them. The reason why I pointed to the Th17 suppression is really based on the in vivo data, because when we take away the KLF2 cells, we find a reciprocal increase in commensal specific Th17 inflammation, but we don't see a comparable reciprocal increase in commensal specific Th1 or other types of effector lineages. So I think in vivo, that data suggests that they're more they're in check to prevent aberrant Th17 inflammation.
Judy, back to you.
Okay, thank you. Thank you very much. So, Alessia, we now have a question from Haidao Shang, who compliments your very impressive talk and asks, why is RNAi in Candida albicans connected, or is RNAi in Candida albicans connected with heterochromatin formation, which is the way we usually think about it? Are there any reports? And in the wild type SC5314 strain, which is RNAi inactive, is heterochromatin still functional as well? And if it functions well, what do you think will be the additional factors and systems that promote heterochromatin formation?
Yeah, thanks for the question. Yes, a great question, and it's exactly where we are thinking to go. So just some background in Candida, in contrast to other organisms where RNAi is linked to heterochromatin, Candida lacks this type of heterochromatin, and this is the heterochromatin with the lysine methylation and HP1 binding. Candida has a more Strelitzia type of heterochromatin that is depending on CIRT2. Having said that, this does not exclude that the two things are connected, and simply because we actually shown, us and Julie's lab actually shown some time ago, that the TLO genes can be regulated by heterochromatin formation as well. Of course, all this study, at the time we didn't know, we done everything in a C5 and so the answer to your question is we do not know whether RNAi and heterochromatin are linked in C. albicans. And the reality is we have to repeat all the analysis that we've done in SC5314 in the clinical isolate that they are RNAi active. But it's exactly what we are doing now. Thank you.
Okay, I think actually I would highlight the point that maybe SC5314, although it's the lab strain, is not at all a wild type strain and has quite a number of different things and genes that are different from the vast majority of the wild strains.
Yeah, I agree. I mean, this I think is one example of many other highlighting that, I mean, perhaps as a community in general, using a reference strain, one strain as a reference, it can never be perfect. So I think we are starting to think in the lab that we start to use a panel of strains. Mm-hmm. And perhaps that's the way to go. It's more laborious, but now with CRISPR it's possible. So I think that's probably the future.
Okay, thank you very much, Alessia. Christina, back to you.
Sing, we got a couple of questions really interested in the role of KLF2 cells in differentiating commensal forms of Candida. Either can they differentiate commensal versus pathogenic Candida, or can they differentiate, can they contribute to tolerance by recognizing only the kind of maternally transferred Candida and differentiate kind of other than when Candida maybe would expand in offspring?
Yeah, no, thanks for that, Christina, and whoever asked that. I probably should have said this or made this more clear, but even though we identified the KLF2 subset of CD4 cells through mice that were colonized with recombinant Candida, turns out that they're not restricted to only Candida colonization, because if we tracked, so once we found that information out and we tracked how CD4 cells respond to other commensals, including bacterial commensals, then a really high proportion of them also expressed KLF2. So I don't think the KLF2 differentiation program is specific to fungal colonization. It's just that we use fungi to find it. But I think it's more broadly applicable to a variety of different commensal microbes. And that's why we think that if it's broadly applicable to suppressing inflammation from fungal and bacterial commensals, then maybe it would have a broader extension to also mediating tolerance to fetal antigens in the context of pregnancy as another subset of genetically foreign good antigens that our bodies need to be tolerant to. With regards to differentiating between commensal and pathogenic, I think that's a really great question. And this is I hope I'm okay to say this, come to our lab in Cincinnati and we can work on this together. We have tons of space, fungi, and funding to do this. So I invite whoever asked that to contact me directly.
We got a copy of all the questions.
Okay, thank you.
Okay. Yeah. So the next question for Alessia is from Derek Sullivan, who's one of the people who's worked on TLO genes, and he compliments your great talk. I agree. A question for her is, does the AGO plus version of SC5314, the one that now is able to do RNAi, differ in cell morphology or growth rate or any other phenotype for that matter in comparison to the AGO minus parent, any in vitro phenotype that is?
Yeah, thanks. And hi, Derek. I hope you're okay. So the answer to the question is no, actually. So when we look at phenotype in YPD at 30 degree, we don't really see much of a difference. I mean, if you ask Elise, the postdoc working in the lab, she says she can recognize the strain, but she cannot, by looking at it, but she cannot pinpoint what the difference is. And this is why we started to think at looking at interaction with the host. So and then I think it's particularly interesting, the thinking that it does alter commensalism in albicans. So what we're thinking now is that because RNAi is an adaptive mechanism, that we need to start to look of how the RNAi mutant responds to host-imposed stress. That's our thinking. And actually, if you think about RNAi in other organisms, perhaps this is not really surprising. RNAi is not essential in the majority of organisms, and it's really there to drive adaptation. And so that's the way we are thinking now about that. But yes, we do.
Okay, thank you. Christina?
Yes, I have another question, Sing from Paul Fidel, who's asked, have you used either IL-17R or IL-22 knockout mice and abrogated the protection against systemic Candida or staph lethal infection following Candida colonization?
Yeah. Thank you for that question, Paul. We haven't specifically targeted IL-17 receptor or IL-22, but we have targeted IL-17 cytokine specifically, so instead of the receptor. And so if we take Candida colonized mice that are then resistant to systemic infection, and we neutralize simultaneously IL-17A and IL-17F in vivo, then we can overturn that colonization-induced protection. And I should have said this too, when they have heightened Th17 inflammation, they also have neutrophilia. And so when you block the 17A and 17F, that systemic or peripheral neutrophilia also goes away. Thank you.
Okay, so we have a question for Alessia from Neil Gao, saying, when you look at hundreds of strains of other pathogenic fungal species, do you also see a mix of strains that have wild type and mutant AGO Argonaut genes? Do you see that in other species?
Yes. So we have not done it for all the fungal pathogenic species you can imagine, but we have looked in more detail at perhaps Lausis, Tropicalis, and Dubliensis. And we had taken the Argonaut sequence and Dicer sequence for all the genomes that are available in CBI for those sequences. And it looks like that they are all RNAi positive. So for the Candida clade that has an active RNAi pathway that has HAGO and ISER genes, albicans seems to be special. Of course, we can't exclude that there is an isolate out there. We also looked at something else. Some other Saccharomyces species have an algorithm pseudogene. And so we looked at the genome of those to see whether any of them were actually RNAi active, and they seem to be all RNAi inactive. So as far as we can see so far, it doesn't look like that we see difference in isolating different species. But of course, I can't exclude it, right?
Right, of course. Okay, Christina?
Yes. Sing Sing, the next question is from Shirley Cohen, who asks, who comments just excellent talk and asks, do you think that transcriptional and morphological heterogeneity within the population of fungi is important for inducing immune tolerance and protection? Or is it more important to have oscillation within an individual cell? Kind of, I guess, reflecting back on the oscillation maybe you were talking about with UME6.
Yeah. Shirley, thanks so much for asking that. No, I think it's really the oscillation, because if we co-colonize with both UME6 on and UME6 off, constitutively on and UME6 null strains, we don't see that systemic Th17 immunity. And so that would, I think, reflect the extremes of heterogeneity, at least with regards to UME6 expression by Candida. But if we force them to oscillate the UME6 using a tetracycline inducible sort of on-off system, then that's where we see it. And turns out during wild type Candida colonization, we can see sort of dynamic states of UME6 on and off too. So I think it's more the oscillating expression as opposed to just heterogeneity in morphology.
While we're talking about the kind of transcriptional topic, I was just curious to know if in these KLF2, you were talking about single cell data, if you get any clues about like a tolerance transcriptional program, if there's kind of broader kind of other genes that are kind of either changes in these T cells across some of these types that give you clues about what is going on in the subset of cells that have KLF2 that, give you clues about what is the tolerance program.
Yeah. Thanks for asking that, Christina. We're really interested in figuring out how, quote unquote, KLF2 works. I think KLF2 had traditionally been known to be a transcription factor that controlled T cell migration into target tissues and controlled hyporesponsiveness of T cells. So our finding that they also mediate commensal tolerance was not totally out of left field, but also added a new dimension for what this transcriptional regulator can do. I would say there's many gene expression changes that are different between KLF2 cells and other traditional effector lineages like Th17s and even FOXP3. But I would say by far the most important change that's different is the production of IL-10 with regards to suppressing colitis.
Gotcha.
And so we're interested to know if that IL-10 production is also important for other things like reproductive tolerance.
Gotcha. Thank you. Judy?
Yeah. Okay. So we have another question from Priyesh Singh, Alessia, plus a combination with something I wanted to ask you. Some of the TLO genes are virulence factors. So it's kind of counterintuitive that you're getting them silenced by RNAi and clinical isolates. And so the question is really more about like, what does this tell you about virulence? And on the other hand, I was thinking of it the other way, which is that SC5314 tends to be more virulent than most of the non-lab strains. And so I'm wondering if you've thought about that and thought about why that might be.
Yeah, I think really the answer to both questions, I think the key here, it's modulation, is fine-tuning of TLO genes regulation. So when I say they are silencing by RNAi, it doesn't mean that they are off in wild type. They are expressed less than in RNAi mutants. Now, TLO genes are a bit funny, and I mean, Derek in the audience will know more than me. They're a bit funny in the sense that they tend to be stoichiometrically higher in amount than the amount of that is required to be in the mediator complex. So with the situation, we have lots of TLO protein, one mediator complex, and the TLO protein are there, and they change the flavor of the mediators. So there's an excess of TLO proteins. So I think the answer to this question is really, what is important is modulate the relative amount, is the relative amount of one compared to another, and the fine tuning. And so this is an answer to both your question, Judy, and the question from the audience. And I just want to make clear here, we are not in a situation where it's on and off. It's higher transcription of lower RNA levels.
Thank you.
You're welcome.
Tina?
Yes. Sing, we have a question from Salome Labgut. Are the KLF2 T cells induced in other barrier tissues and regulate microbiota-induced pathology in those other barriers as well?
Yeah, no, really good question. We haven't seen, in the absence of KLF2, pathological inflammation in other tissues like the lung, for example. I'm not sure, though, that doesn't mean it's not regulating inflammation there, because the density of commensal microbes in the intestine compared with the lung or even the upper airway is quite different. And so is it because our ability to use histology to pick up changes in other barrier tissues is not as sensitive as in the intestine, or is it that it's selective for the intestine? I don't think we know that yet, but clearly the tissue that has the most impressive phenotype is the small and large intestine when mice don't have KLF2 positive CD4 cells. But if you eliminate the microbiota, then that intestinal inflammation goes away.
Okay. I have one more question for Alessia, and that is, you said that all of the TLO genes are just kind of downregulated or upregulated. And I know that they're very repetitive genes, so it's sometimes hard to tell which ones are regulated more. But have you looked at, there's one TLO gene which is actually not at the telomeres, it's inside the chromosome. And I'm wondering if you've seen whether that TLO gene is affected by the RNAi system or not, because one could imagine that its level stays the same and that it's basically the major thing that's running things in the mediator complex, which the TLO protein is a component of, rather than the guys out at the telomeres.
Yeah, you're right, Judy. And actually, especially when you do small RNA sequencing, we get these very small RNA tags. It's actually very difficult to know from where the small RNA come from which TLO the small RNA come from. So we are very careful in not making major conclusion of which TLO is regulated. I use them at the moment altogether. I don't think they're all targeted by RNAi, but we just cannot. TLO34 seems to be target of RNAi. Actually, what I particularly like of TLO34, there is an overlapping of the open reading frame inside TLO34, antisense of TLO34 that could generate the double-strand RNA trigger.
Oh, okay.
So we are now really testing this hypothesis by deleting only just TLO34 and see whether we destroy the whole RNA response. And both Derek Sullivan and Gary Moran lab, but now have also constructed this great strain that has all the TLO genes deleted. So we wanted to use this strain to then put back individual TLOs and see if and when we trigger again RNAi. To really, because I think one of the questions really is, what is the trigger? Where the double-strand RNA comes from? Is this, as you mentioned, is the subtelomeric position? Is there something special about TLOs? The whole question that we would like to really address, and we don't know the answer to.
That's a great set of experimental plans, and actually, it would also be a great strain to look at what other phenotypes are showing up when TLO, the major one of the TLO RNAi, is no longer relevant.
Exactly. Right? That's exactly the kind of thing that it's really, now we want to really have improvement that exists. We really need to dissect and understand what the triggers are. And that's also why I go back to the other Candida species, because Dubliensis has just 2 TLOs. Other Candida have no TLOs at all. So the question there is really, is RNAi there triggered or not? And if it's not, is still a subtelomere or not? And so on and so forth. So that's why I think using different Candida species can be very powerful to answer these questions.
Right. Great. Thank you very much.
You're welcome. Thank you.
Okay. Christina?
Looks like we have wrapped up all of our questions.
Yeah, I think so. So thank you very much to the speakers then, and for a very good session, and to all those who asked questions for making it also a very stimulating question and answer session.
Thank you so much, everybody, for this opportunity. And it's great to be able to talk about our work on fungi, because like I said, I usually don't get to talk about that.
Thank you, everybody. I do talk a lot about fungi, but it's always nice to talk about it.
Well, thank you both, Alessia and Sing, for your talks, and thank you also to the MRC, CMM staff behind the scenes for all their help running MycoTalks. We all look forward to seeing you on the 28th of March, when our speakers will be Blake Billmeyer from the University of Georgia, USA, and Tonis Rokas from Vanderbilt University, USA. Bye, everyone.
Bye-bye.